In this video, we will learn how to download Genome FASTA and GTF annotation files using Conda and the NCBI datasets command line tool. We will use two approaches. First, downloading Genome using a GCF accession number. Second, downloading Genome using a species name, such as Aedes aegypti. First, we use the GCF accession method. We create a Conda environment and activate it. Then we download the genome using a specific GCF accession number. This approach is very accurate because each accession represents one exact genome assembly. We include both Genome FASTA and GTF annotation files, unzip the downloaded file, and copy the genomic.fna and genomic.gtf files for further analysis. Next, we use the species name method. Instead of an accession number, we specify the organism name, for example Aedes aegypti. We again create and activate our Conda environment, then download the reference genome using the taxon command. This method is simpler and useful for beginners, while the accession method is best for reproducible research. Next we will see all the commands using the terminal in practical. I am in the terminal and starting the practical workflow. First, I create a new Conda environment called NCBI_download. This environment will contain the NCBI datasets command line tool and unzip utility required to download Genome and GTF files. I run the command. Conda create NCBI_download C Conda forge NCBI datasets CLI unzip Y. Now Conda is connecting to Conda forge and collecting package metadata. Here we can see the list of packages that will be installed. The important packages are NCBI datasets CLI, used to download genome data from NCBI. Unzip, used to extract compressed genome files. The installation is now complete. Conda suggests how to activate this environment using Conda activate NCBI_download. This means all further commands will run inside this isolated environment. Now that the environment is ready. Next, I verify whether the datasets command line tool is properly installed. I run the command. Which datasets? This shows the path of the datasets executable inside the Conda environment. Now I check the version of the datasets tool. I run datasets version. If the installation is correct, it will display the current version of the NCBI datasets CLI. Now that the datasets tool is installed and working, the next step is to download a genome. First, I open the NCBI genome browser and search for the organism of interest. Here I am selecting Aedes aegypti. NCBI shows multiple genome assemblies. Each assembly has a unique GCF accession number. This accession is important because it represents one specific genome version and ensures reproducibility. I copy the GCF accession number from the NCBI page. Now I return to the terminal and run the datasets download command. This command downloads both the Genome FASTA file and the gene annotation GTF file. We can see the progress bar as the genome is being downloaded. Once the download completes, the file is saved as genome_eats.zip. Next, I unzip the downloaded file. Unzip genome_eats.zip. Inside the extracted folder, we can see multiple files including genomic.fna and genomic.gtf. The genomic.fna file contains the full genome DNA sequence. The genomic.gtf file contains gene annotation information such as gene locations, exons, and transcripts. These files are now ready for downstream analysis such as RNA-seq alignment, genome indexing, and functional genomics workflows. Now I will demonstrate the second method to download genome and annotation files, this time using the species name instead of the GCF accession. First, I create a new Conda environment specifically for species-based downloads and install the NCBI datasets tool along with unzip. After installation, I activate the environment using Conda activate NCBI_species. Environment got activated. Remove any previous files or directory of NCBI_datasets. Now I move to the terminal and run the datasets command. I open the NCBI Genome browser and search for the species Aedes aegypti. I directly use the species name for downloading. Run the datasets command. This command automatically downloads the reference genome assembly along with the gene annotation file. We can see the genome package being downloaded successfully. Next, I unzip the downloaded file. Unzip Aedes_genome_download.zip. If a prompt appears for replacing files like readme, you can press A to overwrite all or choose N to skip. After extraction, I navigate into the NCBI_dataset folder and then into the data directory. Inside this folder, we can see the Genome FASTA file and Genomic.gtf annotation file. Let's quickly summarize what we learned in this tutorial. First, we created our Conda environment and installed the NCBI datasets command line tool. Then, we verified the installation using the datasets version command. After that, we downloaded Genome and GTF annotation files using a GCF accession number directly from NCBI. Next, we performed the same genome download using the species name, such as Aedes aegypti, which is useful when accession numbers are not known. We then extracted the downloaded files and explored key outputs like the Genomic FASTA file and the Genomic GTF. Thank you for watching this tutorial. If this helped you, please like the video, share it with others learning bioinformatics, and subscribe for more practical terminal-based tutorials.